हेलो गाइज, आप लोग कैसे हो। मैं अजीत प्रताप यादव आप लोगों के लिए नई वीडियो लेके आ गया। आज इस वीडियो में डिस्कस करूंगा क्लास 10th का हेरिडिटरी चैप्टर। तो गाइज ये वीडियो क्या है, ये डिस्क्लेमर दे देता हूँ। ये वीडियो क्या है, एजुकेशनल पर्पज से बनाई गई है। तो लेट्स स्टार्ट। व्हाट इज हेरिडिटी? हेरिडिटी इज द पासिंग ऑफ जेनेटिक ट्रेट फ्रॉम पेरेंट टू ऑफस्प्रिंग। इट्स कॉल्ड जेनेटिक्स। Hereditary heredity is the passing of genetic trait from parent to offspring. Who is considered the father of genetics? Gregor Mendel is considered as the father of genetics. What organism did Mendel use for this experiment? The garden pea, Pisum sativum. What is gene? A gene is a unit of heredity, a section of DNA that code for a specific trait. What is gene? A gene is a unit of heredity, a section of DNA that code a specific trait. What is allele? Allele is a specific alternative form of gene. Example, allele of tall versus short plant. Now, question number 6. Differentiate between genotype and a phenotype. Genotype is genetic makeup. Example, capital TT is capital T, small t, small tt. Phenotype is the physical expression of the genotype, tall, short. Genotype genetic makeup. Example, capital TT is capital T, small tt. Phenotype is physical expression of genotype, tall, short. Now question number 7. What does it mean for an organism to be homozygous for trait? It has two identical alleles for that gene. Example, TT, capital TT or small tt. What does it mean for an organism to be homozygous for trait? It has two identical alleles for that gene. Example, capital TT or small tt. Question number: what does it mean for organism to be heterozygous for trait? It has two different alleles for that genes. Example, capital T, small t. What is a dominant allele? An allele whose trait is physically expressed when even when only one copy is present. It is represented by a capital letter T for tall. What is recessive alleles? An allele whose trait is only physically expressed when two copies are present. It is represented by a lowercase example T for small t for short. What is a recessive alleles? An allele whose trait is only physically expressed when the two copies are present. It is represented by lowercase example T for short, small t for short. State Mendel law of segregation. The two alleles for a heritable character segregate separate from each other during gamete formation so that each gamete carries only one allele for each genes. Question number 11. State Mendel's law of segregation. The two alleles for a heritable character segregate separate from each other during gamete formation so that each gamete carries only one allele for each gene. Question number 12. During what process does the law of segregation occur? Anaphase 1, meiosis. During question number 12. During what process does the law of segregation occur? Anaphase 1 of meiosis. What is Punnett square used for? To predict the genotype and phenotype ratio of the offspring from a genetic cross. Question number 14. In a cross between two heterozygous capital T, small t, tall plant, what is the phenotype ratio of offspring? 3 tall, 1 short. Question number: what is the genotype ratio for the cross above capital T, small t and capital small t? 1 tt, 2 tt, 1 tt, small t. 1 capital T to 2. Second is capital T, small t, and 1 small t. Question number 6. What is the test cross and why is it used? The test cross is breeding an individual of unknown genotype which show the dominant phenotype with a homozygous recessive individual. It is used to determine the unknown genotype. Question number 16. What is the what is test cross and why is it used? A test cross is breeding an individual of unknown genotype which show the dominant phenotype with a homozygous recessive individual. It is used to determine the unknown genotype. Question number 17. If an organism with the dominant phenotype is crossed with the homozygous recessive organism and all offspring show dominant phenotype, what was the genotype of the dominant parent? Homozygous dominant. Example capital T. Question number 18. State Mendel's law of independent assortment. Gene for different traits assort independently of one another during the gamete formation. Question number 18. State Mendel's law of independent assortment. Gene for different traits assort independently of one another during gamete formation. Question number 19. When does the law of independent assortment occur? Metaphase 1 of meiosis. When homozygous chromosomes line up randomly at the metaphase plate. Question number 20. What is the dihybrid cross? A cross that examines the inheritance of two different traits simultaneously. Question number 21. What is the expected phenotype ratio for a dihybrid cross between two heterozygous individuals? Capital R, capital Y, small y multiplied by capital R, capital Y, small y. 9 is to 3 is to 3 is to 1. In 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green. Define true breeding. Organism that when self-pollinated produce offspring identical to themselves for a specific trait. They are homozygous. Question number 23. What are the parental generation in its genetic cross? The P generation are original parents. What is the F1 generation? The first filial generation, the offspring of the P generation. Question number 20. What is the F2 generation? The second filial generation, that offspring resulting from a cross between the F1 individuals. Question number 27. Mendel's mean why did the recessive trait disappear in the F1 generation and reappear in the F2? The recessive alleles was masked by the dominant alleles. In the F1 heterozygotes, it segregates during the gamete formation in the F1, allowing it to express again in homozygous individual in F2. Question number 27. What is a monohybrid cross? A cross that examines the inheritance of only one specific trait. Question number 28. If homozygous dominant plant is crossed with a heterozygous plant, what percentage of offspring will be homozygous? 50%. All offspring will have the dominant phenotype, but half will be homozygous, capital T, and half heterozygous, small capital D, small t. What is pedigree? A chart that shows the relationship between a family and the inheritance of the specific genetic trait across the generation. Question number 30. In pedigree, what does a square represent? A circle. A square represents the male, a circle represents a female. In pedigree, what type of square represent a circle? A square represents a male, a circle represents a female. Question number now move to part 2 beyond the Mendelian genetics. Question number 31 to 50. What is incomplete dominance? A blending pattern where the heterozygous phenotype is intermediate between the two homozygous phenotypes. Example, red flower multiplied by white flower equal to pink flower. Question number 32. Give an example of incomplete dominance. Snapdragon or 4 o'clock flower color. What is 30? Question number 33. What is the codominance? A pattern where both alleles are fully and separately expressed in heterozygous phenotypes. Question number 34. 4. Give an example of codominance. ABO blood group, blood type I, A and IB alleles are codominant. R a row roan cow has both red and white hair. Why are multiple alleles? What are multiple alleles? When a gene has more than two possible alleles found in a population through an individual still only has two. Question number 36. Give an example of trait controlled by multiple alleles. Human ABO blood type 1A, 1BI. What is the genotype for a person with type AB blood? What is the genotype for a person with type O blood? 2. What is polygenetic trait? A trait controlled by two or more genes. These traits typically show a continuous range of variation. Give example of polygenic trait in human. Height, skin color, hair color, eye color are the example of polygenetic trait in human. Question number 41. How is the environmental linked to the gene? Environmental factor like temperature, nutrition, and sunlight can influence how gene are expressed. Example: hydro hydro give flower color depend on soil pH. Hydrangea. Question number 14. How is the environmental linked to gene expression? Environmental factor like temperature, nutrition, and sunlight can influence how genes are expressed. Example: hydrangea flower color depend on soil pH. What are linked genes? Genes that are located close together on the same chromosome and tend to be inherited together is called linked gene. Question number 43. Why do linked genes not follow Mendel's law of independent assortment? Because they are on the same chromosome and are physically connected. Question number 44. What process can separate linked genes? Crossing over recombination during prophase 1 of meiosis. Question number 45. What is a sex-linked gene? A gene located on either the X or Y chromosome is called sex-linked gene. What is a sex-linked gene? A gene located on either the X or Y chromosome. Question number 46. Why are X-linked recessive disorder more common in male? Male have only one X chromosome. If they are inherited only recessive allele on their X chromosome, they will have the disorder. Female have two X chromosomes and would need to receive alleles to be affected. Question number 47. Name two common X-linked recessive disorder: color blindness and hemophilia. If a if a carrier female XHPS for hemophilia has a child with normal male XHY, what is the probability of a son will have hemophilia? 50%. What is epistasis? The phenomenon where the expression of one gene is affected by another gene is called epistasis. Epistasis. A phenomenon where the expression of one gene is affected by another gene is called epistasis. Question number 50. Give an example of epistasis. Coat color in Labrador retrievers where one gene determines pigment B black, B brown. Another gene E upon E determines the pigment is deposited in fur. Now move to part 3. Chromosomal basis of inherited. Question 51 to 70. What are chromosomes made of? DNA and protein. Question number 52. What are autosomes? Chromosomes that are not sex chromosomes. Humans have 22 pair of autosomes. Question number 52. What is autosome? Chromosomes that are not sex chromosomes. Humans have 22 pair of autosomes. Question number 53. What are sex chromosomes? Chromosomes that determine the sex of an individual human. X and Y are the sex chromosomes. XX for female, XY for male. What are sex chromosomes? Chromosomes that determine the sex of an individual in human X and Y. X as female, XY male. Question number 54. What is the difference between chromosome and chromatid? A chromosome is a long continuous DNA molecule. A chromatid is one half of duplicated chromosome joined to its sister chromatid at the centromere. Now, question number 55. What is karyotype? A picture of individual chromosomes organized in pair by size. Question number 50. What is karyotype? A picture of individual chromosome organized in pair of size. Question number 56. What can a karyotype be used to detect chromosomal abnormalities such as aneuploidy? Example Down syndrome. Question number 57. What is the mon, non-disjunction at the failure of chromosome to separate properly during meiosis in a phase 1 or 2? Question number Question number 57. What is non-disjunction? The failure of chromosome to separate properly during meiosis in a phase 1 or 2. Question number 58. What is the result of non-disjunction? Gamete with an abnormal number of chromosomes leading to zygote with monosomy or trisomy. What is trisomy? 21. 59. What is trisomy? 21. Down syndrome caused by an extra copy of chromosome 21. Question number what is deletion mutation? When a segment of chromosome is lost is called deletion mutation. Question number 61. What is the duplication mutation? When a segment of chromosome is repeated is called duplication mutation. Question number 60. What is the inversion mutation? When a segment of chromosome is reversed. Question number 6. What is translocation? When a segment of one chromosome break off and attach to the non-homozygous chromosome. Question number 62. What is the translocation? When a segment of one chromosome break off and attach to the non-homozygous chromosome. Question number 64. Who discovered the chromosomal theory of inheritance? Walter Sutton and Theodore Boveri. Question number 65. What does chromosomal theory of inheritance state? Genes are located on chromosome and the behavior of chromosome during meiosis account for Mendel's law. Question number 66. What is a wild type of allele? The allele most commonly found in nature, the normal version. Question number 67. What is the mutant alleles? A rare altered version of gene often resulting in a variant phenotype. Question number 68. What is a bar body? An inactivated X chromosome in a female somatic cells, which appear as a dense, darkly staining body in the nucleus. Question number 69. Why does X inactivation occur in female mammals? To achieve dosage compensation, ensuring that female XX do not have twice the gene expression from X-linked gene as a male XY. Question number 70. What is the genetic recombination? The production of offspring with combination of trait and differ from those found in the either parent, often due to the crossing over. Now move to part 4, molecular genetics and DNA. Question number 71 to 90. What does DNA stand for? Deoxyribose nucleic acid. Question number 72. What is the structure of DNA? A double helix composed of two antiparallel strands of nucleotides. Question number 73. What are the three components of the nucleotide? A deoxyribose sugar, a phosphate group, and a nitrogenous base. What are the three components of nucleotide? A deoxyribose sugar, a phosphate group, and a nitrogenous base. Name the four nitrogenous bases in DNA: adenine, thymine, guanine, and cytosine. Adenine, thymine, guanine, and cytosine. Adenine A, thymine T, guanine G, cytosine C. Question number 75. How does the base pair in DNA? How do the base pair in DNA? With T via 2 hydrogen bond and G with C via 3 hydrogen bond. What is the central dogma of molecular biology? What? DNA, RNA, protein. Question number 70. What is replication? The process by which the cell make an identical copy of its DNA. What is transcription? The synthesis of an RNA molecule from DNA template. Question number 79. What is translation? Question number 79. What is translation? The synthesis of a protein using the genetic code carried by an mRNA molecule. Question number 80. Where does transcription occur in a eukaryotic cells? In the nucleus. Question number 81. Where does translation occur? At the ribosome in the cytoplasm. Question number 82. What is codon? A three nucleotide sequence on mRNA that code for a specific amino acid. Question number 83. What is the start codon? What amino acid does it code for? AUG, which code for methionine. What is the three stop codon? UAA, UAG, UGA. Question number 85. What is the role of transfer RNA? The transfer amino acid to ribosome and match them to the correct mRNA codon. Question number 86. What is a point mutation? A change in a single nucleotide base in the DNA sequence. What is frame shift mutation? An intersection or relation of nucleotide that is not a multiple of 3, which shift the reading frame of the genetic message. Question number 88. Why is a frame shift mutation often more severe than a point mutation? It alters every codon after the mutation completely, changing the amino acid sequence of the protein. Question number 89. What is a silent mutation? A point mutation that changes a codon but does not change the amino acid specific due to the degeneracy of the genetic code. Question number 90. What is substitution mutation? A point mutation where one nucleotide is replaced by another. Now move to part 5, applied and advanced concepts, question 91 to 100. Is plasma a small circular double-stranded DNA molecule that is distinct from a cell chromosomal DNA, often used in genetic engineering. What is a plasmid? A small circular double helix DNA, a small circular double-stranded DNA molecule that is distinct from cells chromosomal DNA, often used in genetic engineering. What are the restriction enzymes? Enzymes that cut DNA at a specific nucleotide sequence. Question number 93. What is gel electrophoresis used for? To separate DNA fragment or other macromolecules based on their size and change. Question number 94. What is PCR? Polymerase chain reaction, a technique used to make a million of copies of a specific DNA segment in a short amount of time. Is called PCR. A technique used to make million of copy of a specific DNA segment in a short amount of time is called PCR, polymerase chain reaction. What is CRISPR? CRISPR. Case 9. A powerful genetic editing technology that allows scientific to alter DNA sequence precisely. What is CRISPR cloud? Case 9. A, bit, a powerful gene editing technology that allows scientists to alter DNA sequence precisely. Question number 96. What is an genetic modification organism? What is genetically modified organism? GMO. Question number 96. What is genetically modified organism? GMO. An organism whose genetic material has been altered using a genetic engineering techniques is called genetically modified organism. An organism whose genetic material has been altered using genetic engineering techniques is called genetically modified organism. Question number 97. What is clone? A genetically identical copy of gene is called clone. A genetically identical copy of gene is called a clone. Question number 98. What is the goal of Human Genome Project? To determine the complete nucleotide sequence of the entire human genome and identify all the genes it contains. Question number 19. What is the sex influence trait? A trait that is spread differently in male and female, but the gene is on an autosome pattern. Baldness. Question number 99. What is a sex influence trait? A trait that is expressed differently in male and female, but the gene is an autosomal. Example pattern. Baldness. Question number 900. What is the difference between a character and a trait? A character is heritable feature. Example flower color. A trait is the variant of character. Purple flower or white flower. तो गाइस ये वीडियो आपको अच्छी लगी तो प्लीज़ लाइक एंड सब्सक्राइब जरूर करें। जय हिंद, जय भारत।